All right, so now we are going to go over some examples about how sensory stimuli produce action potentials. So we've talked about EPSPs, IPSPs, we talked about how an action potential occurs, we've talked about the membrane potentials that promote an action potential. In the next chapter, we're going to be talking about neurotransmitters that are involved in the chemical transmission that moves from one neuron to another neuron. But we have sensory information, and this sensory information comes in through the world. We have bodily sensations, touch, balance, auditory sensations, hearing, sight sensations, chemical sensations such as taste and olfaction. And so how would these sensory stimuli produce action potentials? There are specific neurons that are related to these diverse receptors, and they all have ion channels on their cell membranes. So the basic principles that you've learned in terms of what begins an action potential, such as the influx of sodium ions, this pertains to all action potentials throughout your body. The ion channels, once they open, and even sometimes once they close in the context of recovering from an action potential, they initiate a chain of events, and a opening of sodium channels will eventually result in the production of a nerve impulse, and the opening of potassium to begin to return the cell to its negative self will complete that action potential to return the cell to normal to again begin another nerve impulse. So I'm going to give you one example today of one of the sensory stimuli, and we'll be talking about more of them in later chapters. We'll be covering vision and auditory information in particular depth. And today I'm going to talk a little tiny bit about touch. So each hair on our body allows us to detect the slightest displacement. So the dendrite of a touch neuron is wrapped around the base of each hair. Hair displacement opens stretch-sensitive channels in the dendrite's membrane. When channels open, they allow an influx of the sodium ion sufficient to depolarize the dendrite to its threshold level. So here's an example of this. Okay, so here's your skin, here's your hair. Right, this is just an example that I like this example because it's on your arm, and so your hairs, they are very, very sensitive to displacement. I don't think we sort of think about how sensitive they are to displacement, but I'll give you a couple examples in a second. So when this hair moves, the dendrite of the sensory neuron, okay, here's the dendrites right here, they're wrapped around the hair. So when the hair moves, this causes stretch-sensitive channels. So you can have a channel that, the sodium channels that would open if they receive a particular neurotransmitter. You have voltage-gated. Well, these ones are stretch-sensitive channels. So these stretch-sensitive channels, they correspond to pulling movement that causes the sodium to enter the channel and enter the cell. And then again, this makes the cell more positive. And once it becomes more positive, then you have these the potassium channels open, and they start to let out the potassium ions, which are going to eventually return the cell to be more negative. Meanwhile, these sodium ions diffuse, and they have sufficient positivity to again result in the opening of these voltage-sensitive Na channels that will produce a nerve impulse. Okay, so you have stretch-sensitive channels that are going to cause a graded potential that are going to result in a change in the local potential, the local charge of the area. If this becomes sufficiently positive, then these voltage-sensitive channels will open, and so on and so forth. This will travel down the axon. So to see how this works, try to take your arm, set out your arm, and try to brush your fingers across your arm so that you only with your fingers touch the hairs on your arm. Now try it, really try it. And you will be able to feel, and it sort of feels this sort of weird tickling sensation. You'll be able to feel your hands are not touching your arm, but you'll be able to feel the fact that your fingers are touching the hair cells, touching the hairs that causes displacement, that you can then sense that there's something nearby your arm. So you can imagine how this would be true, this would be advantageous evolutionarily, because anything that's coming in from the environment, it's actually going to touch your hairs before it touches your body. So if you have really sensitive hairs, then if you have an automatic reaction, you can imagine like a mosquito or any sort of bug or something like that, and it's going to land on you. And if you get the sort of advanced notice from the hairs on your arm, then you might be able to jerk away or possibly even swat the bug so that you can prevent this foreign body from affecting you. To give you some other examples of how these hair cells work and how sensitive they are, think about if someone were to play with your hair, how it feels when your hair is cut at the barbershop or hair salon. And to give you another interesting example, think about the pets you have, if you have any pets, and how they like to be petted. They're covered in hair, so you're going to pet the hair on their body. This is a pleasurable sensation for them. So you can stroke one of your pets by just touching the hairs and not actually pressing down enough, register physical touch sensations on their skin. So these hair displacements, we don't often think about them, but we actually use them or rely on them way more than we would realize. Another example is how a nerve impulse produces movement. So spinal motor neurons, they send nerve impulses to synapses on muscle cells, okay? So here, the spinal neurons their motor neurons are going to come down your spine, and these motor neurons are going to end in these muscle fibers at these muscle end plates. This would be an axon, this would be a motor end plate, okay? The axon of each motor neuron makes one or more synapses with these sort of target muscle cells. At the end plate is a part of the muscle membrane that is contracted by the axon terminal. So what that means is that here's a closer example. Here's the end plate, here's the axon terminal, and this is in the muscle fiber. And here's the motor neuron. So acetylcholine is a neurotransmitter. So this acetylcholine is released at this axon terminal. Okay, again, it's neurons send chemical signals to each other. You'll be learning about this in the next chapter. This acetylcholine is released here at the end plate, and neural communication. A neuron is going to hook up with another neuron. Well, in this case, the end of a neuron is hooking up with part of the muscle. Okay, and at this muscle end plate, the neurotransmitter is released, and these transmitter sensitive channels open. So if we have within this muscle fiber, if it receives acetylcholine, then what's going to happen is these transmitter sensitive channels open. Sodium floods in the cell, potassium leaves the cell, and this is going to result in a change in local potential that is going to activate voltage sensitive channels, and if sufficiently activated, then an action potential will occur, and this action potential causes the muscle cell to contract. And so in this contraction, then movement occurs. So here we have this sort of singular muscle cell that has this information that's coming down from motor neurons. So your brain is sending information down your motor nerves to try to cause muscles to contract at the cellular level. So when you engage in movement, you're engaging in finely coordinated release of neurotransmitters and depolarization of neurons to result in action potential that causes muscles to contract at very, very specific places, so that then you are able to do the fine hand movements or any sort of fine body movements that you would need to interact adaptively with your environment. This is actually the end of Chapter 4. This is the last slide. Next chapter, Chapter 5, we'll be going over how neurotransmitters are made, what type of neurotransmitters there are, and how they are used to communicate across neurons.